Welcome to the Salk Talk podcast. I'm your host, Chris Emery. Joining us for this episode of Salk Talk is Shani Stern, a neurophysiologist working in the laboratory of Salk professor Rusty Gage, whose research focuses on understanding the biological underpinnings of mental diseases such as bipolar disorder and schizophrenia. To learn more about the Salk Institute and its research, visit www.salk.edu. Welcome, Shani.
Thank you.
Your career has taken some fascinating twists and turns. Let me see if I have this right. You got your undergraduate degree in electrical engineering and then served in the Israeli Defense Forces. Then you worked as an electrical engineer for Intel and Motorola for several years. Then you earned a doctorate in physics from the Weizmann Institute in Israel. And now here you are at the Salk Institute studying the brain. Is that the right sequence?
Yeah, pretty much. So most people do it the other way around. First, they go to school for a long time, and then they work. But my family, there are a lot of electrical engineers, and I also thought, especially at high school, I liked math, I liked physics, I liked computer science, and I thought it would be a good path for me. So I learned it, and then I had to serve in the army, as all Israelis do. And I served a little bit more because I was an officer. And then when I sent my CV, I got like a million offers because electrical engineers in Israel are very, they're very needed. So it was like a golden cage that, I was earning a good salary. I didn't want to leave because it was a good position. But at some point, I kind of felt that I knew more than anybody when I worked for Motorola in speech. I did speech algorithms, and I had nobody that I could learn from. And I got a little bored. So then I kind of thought of starting to go back to the academia. I also started being interested in biology, which was not during high school, it didn't interest me at all. I got, more mature. I had children. I started having children. I started to read. It sounded really interesting. And so I started being interested in biology. I went first to the regular universities, and they said, you need to make a lot of courses.
I see, because you were an electrical engineer.
Yes.
You didn't have that background.
Yeah, and then I went to the Weizmann Institute, and they interviewed me, and they said, you know what, we really want you here. Don't make up anything, just come. So I came, and I studied so I didn't make up any courses, but I just studied on my own. And I I started in computer science. I did my master's in computer science in the bioinformatics track. So I just learned the biology. And then during my PhD, I really wanted to get even deeper into the biology, do wet lab work. So I joined the lab of Professor Elisha Moses, who is doing physics, but is neurophysics. And when I started in his lab, I kind of did more neurophysics, stimulation with electric and magnetic fields. But what interested me a lot was the biology. So I kind of started to pull towards disorders. And Elisha was fine with it. So that's how I ended up. And I started to collaborate with the Neuroscience department with Menachem Segal. And that's how I ended up studying brain disorders. So.
I see. And you mentioned having children. Was there something about that got you interested in biology?
So first, it's really a miracle, right? So everything, I think that it changes everybody. The first time you have a child, it changes you. And then I really started to read. And once you start to read, you realize it's really interesting. I don't know if they didn't teach it well in high school, or I was just not mature enough to realize how interesting it was, or I don't know.
Yeah, it's. I have a 3-year-old, and it's amazing to watch a child go from being born to being 3 and talking and walking and running around. And
yeah, it is amazing. Yeah.
So you and I actually have something in common because I worked in a lab out of college as a neurophysiology technician. So I have a little bit of a head start on maybe some of our listeners as to what that actually means. So could you tell people a little bit about what is neurophysiology and why is it important in neuroscience?
Yeah. So we actually we have many techniques for looking at the physiology or electrophysiology. But when you really want to record currents and potentials, you go to an old technique, which is a patch clamp, which was invented like back in the 70s. And you generally record, a single cell or 2 cells, not more. And what you do is you have a pipette with an electrode inside, and then it is connected to an amplifier and an A to D, which connects to your computer. And what you do is you go very, very close to the cell. You apply small suction. You rupture the membrane, and now you're fused with the membrane with the cytoplasm. You can record currents and potentials of the cell. And then, you can, just because you're really the resolution is high, you're really connected, the noise is low. You can record with some protocols specific types of currents or ion channels. And this is when you study disorders, and the changes are usually they're small because these are functioning organisms, right? They're just a little bit different, and the changes are going to be small. So you need this resolution. You need to measure these small currents, these changes in the small currents, to see the differences.
So your work in the lab most recently is focused on bipolar disorder, which used to be called manic depression, I think, in the past, but
yes.
it's since been renamed. This is a disease that can cause really terrible disruptions in people's lives. And do we know what's actually going on in the brain during these different phases? What's actually malfunctioning?
So that's where our modeling system comes, because it's hard to record from actual brains. But we have the IPS technology.
It's induced pluripotent stem cells.
Yes. And we can differentiate them into specific types of neurons. So we can look at kind of different areas of the brain and see how the neurons react differently. So, for example, we looked already at several types, but the one that we have the most statistics for is the dentate gyrus granule neurons. This is an area in the hippocampus which produces newborn neurons. And we see that bipolar neurons are hyperexcitable. They produce more activity, and they have sustained activity. So they can, for example, if I inject a current for a control cell, it will fire, and at some point it will just get tired and stop firing. But the bipolar cell can go on and on, just firing without any rest.
So the cellular version of mania.
Yes, yeah. And then we're starting now to do more types of cells. So we have a lot of data from CA3 neurons. It's also hippocampal neurons, and we see, for example, that only the bipolar patients that respond to lithium have hyperexcitability in the CA3 cells, but the ones that do not respond to lithium do not have hyperexcitability. But their changes, for example, are in the morphology of the cells. They just make huge CA3 cells, the bipolar non-responsive cells.
And lithium it's the common treatment prescribed for people with bipolar. Yeah.
So usually it's the drug that psychiatrists would start with. It's kind of a miracle drug because it helps in both stages, both in the phase of the mania and the phase of the depression. But it works well only about 30% of the patients. And then 70% of the patients, some of them do not respond at all, some of them respond not well enough. But it's being prescribed to everybody, or as the first treatment, line of treatment for bipolar disorder.
I see, even though there's this fairly large percentage that are non-responsive.
Yeah, so it's kind of like the drug that most psychiatrists would think of first with bipolar or associate with bipolar disorder is lithium.
And you just recently published some findings that may help explain why some people respond and some people don't. Can you tell me a little bit about that?
Yeah, so in the dentate gyrus granule neurons that we did the study,
the dentate gyrus is a portion of the brain,
of the hippocampus.
The
yes.
hippocampus. Yep.
And we see that So all the bipolar dentate gyrus granule neurons are hyperexcitable, or on average are hyperexcitable. But we see that they are very different if they come from patients who respond to lithium and patients who do not respond to lithium. So the kind of only thing that they share in common is hyperexcitability, but they have like very different characteristics or electrophysiological characteristics. Just as an example, if we look at the spike height, lithium responders have it higher than the control on average, or like statistically significant, but non-responders would have it lower than the control. So the spike shape is different, the way that they fire is different. So we were able to predict, based on electrophysiological features, whether the patient would respond to lithium. So we don't even treat the cells with lithium. Also, although if we do treat these cells with lithium, we see that the lithium responder neurons would decrease hyperexcitability, while the non-responders do not. But the way we predicted was not even with treatment with lithium, because just their intrinsic properties are so different that we didn't even need to treat with lithium to find out who's going to respond to lithium and who will not. So generally, what we do is we had 6 patients. So we take, and 3 of them are lithium responders and 3 of them are non-responders. So we take 1 patient out of the sample, we train a model, a naive Bayes model, by the five patients, the electrophysiological features, and then we predict or classify the 6th according to the model that was built by the 5. And we do this iteratively, so each time we take a different patient out, and this way we can assess the performance of the classifier, and we get an error rate which is lower than 8%. For prediction.
And so does that mean that there may be ways to look at these the neurons of or the skin cells of a person with bipolar and actually predict whether they should be even given this treatment or not?
Yes,
that's the idea eventually, or hopefully.
yeah. So we're now trying. So right now the method is you take skin cells or blood cells. You make them into induced pluripotent stem cells, then into neurons, and then measure 5 neurons. That's all you need. From 5 neurons, which is, for me and as an electrophysiologist, it would take me about 1.5 hours to measure. From these 5 neurons, you can predict with very high accuracy whether this patient would respond to lithium or not. We're now trying to make these prediction even more easy, just by doing some other methods which might be easier. We're working on it right now.
And there's I think you told me before when we were talking that there's actually, that's important because there's a downside to being prescribed these drugs if you're not going to respond to them in the normal or in the ideal way. Yeah,
so there are two problems. First is that the patients are not getting the treatment that they should be getting. They're getting lithium, which does not affect them. And second, there are many side effects to lithium. So it's bad to treat people who will not respond to it with the lithium. So yes.
So are there other, you've clearly pursued the neurosciences. Are there other questions about the brain that are just burning questions for you?
So there are always questions, yeah. Right now I'm kind of focused on psychiatric disorders. This is how, I think that's the main reason I went back to the academia. Disorders or disease was interesting for me, but I am, always also interested in the basic science. Right now, as an electrophysiologist, I'm helping other people in the lab, which sometimes do basic neuroscience, to develop new protocols for other types of cells, for example.
Dr. Gage's laboratory studies a number of disorders, including schizophrenia. Are there any connections between bipolar and schizophrenia?
Yeah, so it's kind of like I did a lot of patch clamping from schizophrenia. And generally what we see is that they're hypoexcitable versus bipolar, which are hyperexcitable. So, but since if you look at the behavior, sometimes they have some similarities, right? So we're not sure how the behavior, is how this is translated to behavior. But the only thing that, I did a lot of schizophrenia lines, helping with other people's projects, and their cells are hypoexcitable, which is, by the way, most of the disorders are hypoexcitable. I did a lot of Down syndrome before, that it's hypoexcitable, and autism is hypoexcitable. Usually disorders are hypoexcitable neurons. And kind of bipolar, I think it was the only one that we saw. I have to make sure in my mind that that is hyperexcitable.
One thing I'm curious about is because I used to work in a neurophysiology lab, I know that, the techniques that I used, and this was, 20 years ago, the techniques have carried on. Are there any, areas of technological development in neurophysiology and recording the electrical activity of neurons at that level that have evolved recently?
Yeah. So there are many methods that are helping a lot, like multi-electrode arrays, and sometimes there are some voltage-sensitive dyes, so you can see, or calcium imaging. That has evolved, but still, if you want to get to resolutions of specific ion channels, currently the only method is to go and do patch clamp.
So you have 4 children, correct?
Yes, I do.
Makes me tired just saying it.
Yes.
And you're you have this background in math and engineering. Do you what goes on in your household?
Yeah. So, first I learned the first thing I learned very well was to parallel things. So I make dinner and I help with the homework and I'm folding laundry. Everything is in parallel, done in parallel. Second, I teach math, like high level math, to my children. I know it started when they were very, very little, and I thought, like, in the preschools they weren't getting enough. So I kind of started then. It just, it went well. So I continued, and now, like, everybody studies, like, the math that is like a few grades above his level, and they're fine with it usually. So yeah, I'm just continuing with it. I think it's good for them. So yeah.
So they seem to have an interest in science and math. And
They do. Yeah, I think they do.
are there any outliers, or do you have a child who's like completely gone off into the arts?
Well, they do. They like the arts too. So it's not that they're only focused on science. It's just, for me, it's harder. Like, I also like painted when I was younger. Now I don't have time, but I don't have the time to to teach them. And the schools here are very good with the art. So they're learning in school, and they like the art very much.
So how long were you in the military?
3 years.
3 years. Did that have any influence on, switching from being focusing on electrical engineering to biology?
I don't think so. I think that what I did learn just before from being an officer was, just to parallel everything and to delegate authorities. So my children help me a lot. We have one child making breakfast, one child making dinner. We clean the house. Everybody cleans together. Everybody knows exactly their role in which time they need to do it. Sometimes they complain a little bit, but there is no choice. So yeah.
The Stern household is an efficient operation.
Yes,
run
definitely.
by an engineer and former military lieutenant.
Exactly.
Well, Shani, thank you so much for joining us today.
Thank you.
To learn more about the Salk Institute and its research, visit www.salk.edu.